Hello, students. Dr. Dia here, giving you a quick tour through the topic of nucleic acids. Please open your PowerPoint notes, and we're going to start from there. This is our slideshow here, and if you remember, I believe last time at our previous meeting, we discussed the nucleic acid building blocks. This was a lecture I did all on the whiteboard. So today we're going to talk about polynucleotides. Remember that as we've discussed before, the way biological molecules are assembled, you start with certain building blocks which you link up to form the oligo and poly, whether it's, we talked about saccharides, we talked about peptides, and now we're talking about nucleotides. So nucleotides can be connected to form oligonucleotides, and as before with oligopeptides and oligosaccharides, we're going to call this a chain of between 2 to 10 nucleotide residues. Uh, Polynucleotides would be more than 10 nucleotide residues. This is a sample of a poly or oligonucleotide. There are several things to take note of here. For example, you can see that over there on the top we have a 5' phosphate. Remember that we numbered the carbons on the sugar subunit by putting prime numbers on them. And carbon number 1' would be the one where the nitrogen base is attached. We call that an N-glycosidic bond. And then over here we have on the top a phosphate attached at the 5' carbon. Notice that in this case we have a 5' terminal residue would be thymine. Remember that we're going to start counting nucleotides from the 5' towards the 3' end. Over here, we have the 3' terminus. Notice that it's free right now. Also notice that in this particular situation, we've set up a adenine as your 3' terminal residue. Notice if you look at, let me see if I can do something here, just a second. Again, notice that in these particular cases, what we have is a particular configuration of the sugar. You'll notice here for each one of these units here, you'll notice that at the what we call the 2' carbon, I'm trying to use the highlighter here, but I'm not getting it to work. Oh, here we go. Here is the 2' carbon, and you'll notice that what we have there is a hydrogen instead of a hydroxyl group. That's because this particular ribonucleotide is a deoxyribonucleotide. It belongs to the class that we call DNA. So remember that in DNA we use a deoxyribonuclease. I'm sorry, the deoxyribose sugar that doesn't have a hydroxyl at carbon number 2'. All right. Okay, so let's go ahead and take that out. Let's move on to the next one. Now, over here, what I'm showing is this is a ribonucleotide. Notice that it has at the 2' carbon, it has an OH instead of an H. Notice also that instead of the base thymine, it's going to have instead uracil or U. And I'm not making you guys memorize all the different formulas and structures or actually the names. I just want you to remember the letters that represent them. So A, G, C, U, and T. Okay, a few seconds so you can take a look at this. Pause the video if you need to. And let's talk now about DNA structure in particular. As we will review constantly, remember that the idea is nucleic acids, the major function they have is in the storage and transmission of genetic information. And the storage occurs in these molecules of DNA. So we're going to say that in the DNA structure, there are stretches that carry codes for protein production. That is what we're going to call genes. Of course, the term gene and genetics came from studies that were done way before DNA was actually identified as the biomolecule that carries that information. But genetics have been around for a long time, And we knew that there's certain traits were inherited from one generation to the next. And so we now can make a correlation between those traits and specific sequences of DNA that carry a code for production of certain proteins. Um, in our study of proteins, we discussed that there were several levels of organization or structure. The same is true for DNA, and we're going to find that we can understand the structure of DNA in terms of three levels: primary, secondary, and tertiary. Primary structure refers to the sequence of the nucleotide residues in a stretch of DNA. Secondary structure, on the other hand, is very interesting, and it consists of two DNA strands that are held together uh, to one another by base pairing. And base pairing means hydrogen bonding that's going to happen between the bases that are attached to the sugar phosphate backbone. This base pairing is complementary. And the way it works is very exclusive in that uh, the base adenine or A can form hydrogen bonds with thymine or T, and guanine or G can form hydrogen bonds with cytosine or C. And this is very important because, as you can see, when you see the structure, you'll realize that the only way you can fit these bases inside this structure is if they pair this way. This is the only way they can pair, much like a lock and key mechanism, but there's only enough room inside this structure to fit them. This is what we call the double helix. These are the pairings. You can see where on the left here, A can pair with T by forming complementary hydrogen bonds, two of them, and G can pair with C by forming three complementary hydrogen bonds. And these are very specific, and given the structure of DNA, these are the only way that you can pair these guys. And it has to be, again, a purine with a pyrimidine. Remember, the purine is the one that has the double ring. Pyrimidines are the ones that have the one single ring. And that's the only way they can fit. It has to be a purine with a pyrimidine. Otherwise, they will not be able to fit into the DNA's secondary structure. Now, in the most common form that we find, there are two strands and they're lying side by side in an anti-parallel spiral arrangement. One strand of the DNA runs from the 5' towards the 3' end, and the complementary strand runs in the anti-parallel direction from the 3' to the 5' end. They coil around each other in the form of a double helix. And probably the best illustration I can get you of what this means is like a spiral staircase, like this one here. Notice that if you see the handrails, well, that would correspond to the sugar phosphate backbone of the DNA strands, and then the steps would represent the base paired bases inside that double helix. Here is again an illustration, and again it's kind of hard to visualize, but what you can see, let me show you the diagram on the right. It shows the sugar phosphate backbone as a ribbon, and you can see the coiling where one of the strands is moving from the 5' towards the, let me see if I can, oops, sorry, that didn't mean to do that. What I wanted to do was I wanted to see if I could get the highlighter. You can see where one strand here has the 3' end on here, and you can chase it around, and then the other one has the 5' end over there. So you can see they're anti-parallel, and you can see how the bases are in between these two strands. Now, if you could look at this in the form of the whole set of electron clouds, you'll realize that's what's happening is the coiling creates a major groove and then a minor groove on the surface of those coils. And these are the features that proteins are going to recognize when they attach to DNA to perform different kinds of enzymatic or regulatory functions. They're basically going to be fitting into the major or minor groove of the DNA double helix. All right, let me get rid of this highlighter function here and let's move on. Now, One of the things that's important is that the sugar phosphate backbones essentially are going to be what we would call the outside of the double helix. And the reason for that is because the phosphate groups are going to be negatively charged. Remember that phosphate is a derivative of phosphoric acid, a weak acid, which therefore means that at physiological pH, which is what we've defined as pH, 7.3, 7.4, uh, acidic groups are going to be fully ionized, and so they're going to have negative charges on them. And so these negative charges are going to be facing the outwards direction of the double helix, which allows it to remain soluble in an aqueous environment, whereas the bases are going to be on the inside, and base pairing is going to happen there. But besides that, what's going to happen is that the bases of DNA are going to be stacked, and that's going to maximize hydrophobic or nonpolar interactions between neighboring bases, and that's going to stabilize the secondary structure. If I go back to the previous slide, you can see where these bases are kind of stacked there in the middle. You can see them here. And they're basically going to form. Remember that these structures are, if you look at it on the left, they're pretty flat. And so what's going to happen is they're going to be stacked upon each other, and this maximizes the hydrophobic or the London dispersion or nonpolar interactions between these bases. Okay, let's go back here to our. This is an actually assembled three-dimensional model. And again, it accentuates, if you can see all of these reds spheres here, those are the oxygens in the phosphate groups. And as you can see, they're all on the outside, forming like the handles of the spiral staircase. You can probably trace on the outside this major groove that goes around the DNA. And then in the interior, you can see how the bases are stacked, forming those stabilizing hydrophobic interactions with each other that strengthens the structure of the DNA in this form. Now, there is also a tertiary structure. If you can imagine, a double helix of DNA would be like a long string or cord. It would be very hard for it to fit. And the question is, how do we fit these incredibly huge molecules of DNA inside the nucleus of a cell? Well, this is where tertiary structure comes in, and this is the overall three-dimensional shape and includes the secondary structure. Now, there's a couple of interactions that happen here. One of them is a tertiary structure that arises from supercoiling. So, again, I am sorry that I cannot provide you with better illustration of this, and please disregard this comment here about figures 1411. Please disregard that. That's from a previous book that we used to have. We don't have it right now, so disregard that. But this is basically there are enzymes that can take, for example, this is what's called a plasmid. This is a circular molecule of DNA, and many bacteria have these kind of satellite pieces of DNA that are not part of the main genetic information of the bacterium. And what a enzyme called a topoisomerase can do is it can basically put a nick in there and cause this thing to kind of like form a supercoiled or a very tightly wound, basically, piece of DNA. So something like that happens in bigger cells. It's essentially a tighter, more compact shape. And in the case of DNA, when DNA is not actively dividing, what you're going to have is this DNA is going to be twisted around a complex set of proteins called histones. And you can see it here, where if each one of these blue balls that you see here represent a complex of these proteins called histones, the DNA is wrapped around it into a supercoiled form that makes it very compact. It's easier to package. So you can see on the left side here When the DNA of the cell is packaged in a non-dividing scenario, what you see is the DNA wrapped into these strands. But essentially, what it is a complex between DNA and these histone proteins, and we call that chromatin. Call this chromatin. Now, when the cell prepares to divide, what will happen is that chromatin condenses even further into these structures that we call chromosomes. And if you take in a biology course or a genetics course, you'll remember seeing that the human DNA, for example, the DNA will pack into 23 visible chromosomes, or actually pairs of them if it's going to divide, and then they divide into the new cells. Okay, let me get rid of this highlighter here and continue talking. This is an electron a photomicrograph of a dividing chromosome. I believe this might be the so-called X chromosome. So what you're seeing essentially is a structure that consists of enormously long strands of DNA wrapped around these histone complex proteins and forming this structure, which when the cell divides will be transported one copy of each into each of the daughter cells as the cell divides into two. In the lab, we are capable of doing many things. We can actually isolate DNA, and one of the activities we'll do is we will go into our lab and actually isolate DNA from fruits. I think we'll probably do strawberries, but you'll be invited to bring other fruits and hopefully isolate the DNA. Now, when the DNA comes out, it is very viscous. It's very kind of jelly-like. But there are ways that you can use to affect that structure. We call it denaturation. Uh, just like proteins, remember DNA structure is held by hydrophobic interactions and hydrogen bonding interactions. Just like proteins, the same kind of conditions can be used to denature nucleic acids the same way that we previously denatured proteins. So, for example, one of them was heat. If you apply heat to a sample of DNA, you can actually disrupt the double helix and cause it to unwind and actually separate entirely into two individual strands of DNA. If you do this process in a gentle, slow way, you might be able to separate them all the way. And then if you cool them down gently, you might be able to actually reverse the process like it says here. Cooling can reverse the process, essentially renaturing the DNA. This is not always possible. Sometimes, although we don't have time in our course to go through this, uh, DNA can be modified later on by different enzymes. And once you do that, you might not be able to renature the two strands of DNA. But essentially, what's going to happen is because this denatured DNA consists, like you can see, one, two strands, but those strands are complementary to each other. In other words, the sequences of nucleotides on one strand can actually find their complementary sequences on the other one and reform the complete double-stranded DNA. Like I said, this is not always possible, but many times it's very helpful if you want to introduce your own modifications into the DNA. Okay, in our next lecture, we're going to talk about nucleic acids and their role in information flow, and we'll talk about that. So hopefully this lecture, little brief little presentation, has helped us to get a better view of how DNA is structured and packaged. All right, so we'll go here.